how do you control on a population basis any infectious disease, let alone a fatal infectious disease at this point in time? You make a vaccine.
In the case of the HIV vaccine, it's got to totally 100% prevent infection. Or if infection occurs, it has to totally 100% eliminate it before it gets into the genetic material of the host. And that's a very, very high bar.
1953, when Edmund Hillary climbed Mount Everest for the first time, nobody thought it was possible.
And HIV still remains that really mountain of scientific obstacle.
We learned about HIV on the fly. When the virus was first discovered, we initially assumed it was going to be like other viruses that are what I would call monolithic. They don't change.
And it turned out it didn't act like any other virus.
The amount of diversity in influenza, that exists globally is significantly less the genetic diversity of HIV in a single individual. I mean, if you think about that, that just blows your mind.
It was known back then that it integrates into the human immune system as it infects.
When a patient, after getting infected, went past the acute phase, they were completely asymptomatic. So that was very difficult to explain why the disease was progressing. So in our discovery has completely changed the paradigm, then and demonstrating that there was no virologic latency, but continuous active HIV replication.
The virus hid in the center of the lymph node, we call it the germinal centers, where there are no killer cells or CD8 cells. So it was in what we call an immune privileged site. It was like, geez, how clever.
There were a couple of trials with recombinant proteins that failed miserably. That was the warning shot over the bow that we couldn't do business as usual.
And we were particularly focused on CD8 T cells, which are ones that really do recognize infected cells and are really the killer.
That the virus had developed some immune evasion strategies that we had never even thought existed or could conceive of. That it was able to mask the conserved epitopes. That it was able to focus the immune response on a single protein, and it was making many other variations of that protein, so it escaped from it right away. And it was a revelation as to how complicated and how many immune defense mechanisms that the virus was taking out, that the concept that it would create diversionary proteins and that the concept of what we call immunodominance arose out of these, quote, areas of HIV. And the recognition now that it was the subdominant responses that are actually what you need to elicit.
There really was a shift away from trying this cellular immune response approach to recognizing that we probably needed antibodies.
We had hoped that, by combining both T cell responses and antibody responses that we could do better. But that was not the case. There was a very mild signal there that there was some protection in the vaccinees. But unfortunately, when we moved into a different population, we were not able to replicate those results in that population. And so it really also explains why it's important, to look in diverse populations to make sure that your vaccine is going to be effective worldwide, not just in one group of people.
What this did was to provide proof of principle that a vaccine was possible.
Is we would generate many antibodies, but very few of them were neutralizing. And so what the virus had learned to do, probably evolved to do, was to present pieces of its viral envelope to the immune system that it didn't care about, that weren't really critical to its function. And it would do this by, masking the envelope, putting glycans on the surface. It would do this by creating motion of the envelope so that nothing stood still long enough for the immune system to see it.
And our approach is understand the antibodies that work, break down in molecular terms how they've done what they've done, and then transfer that knowledge into the design of molecules, immunogens, vaccines, basically, that will induce those antibodies.
Now we're forced to really do this incredibly deep biology of learning how does the human respond to this complex virus in order to make these antibodies fundamentally the body doesn't want to make.
And then ultimately what we decided was that what we really needed was neutralizing antibodies to provide protection. And so what we think is that we need a combination of broadly neutralizing antibodies to provide more breadth and cover more strains of HIV.
We learned that one antibody is not enough. And we have seen this in treatment for HIV. So that is really, I think, where the next steps are going to be, is combining antibodies, both for passive immunization as well as active immunization, in order to cover HIV's ability to mutate and get around our prevention strategies.
The most recent clinical studies showing that we can guide the immune system to a series of immunizations to start to make really good antibodies and really specific immune responses. Those are very encouraging.
I hope to see that the broadly neutralizing antibodies themselves will provide protection when given in combination. But I also really hope to see that we develop vaccines that could generate broadly neutralizing antibodies, because the goal there is to try to generate a more long-lived approach to providing protection against HIV acquisition.
So I see the pathway to a vaccine. I hope that we can remain committed to it.
Well, yes. Given the progress we are making right now, and given the new tools we can possibly leverage in the modern days, I think someday we will be able to climb, Mount Everest, a daunting task to achieve it. I don't know how long, but I think we can achieve the goal.